All right, so I wanted to make a video for those of you that are not in class this week, and also I'm going to be gone from fourth period today, and so I wanted to make this video quick, highlighting a couple things so you know what you're doing on your assignment, and so you understand how to do a monohybrid cross and how to use a Punnett square. Okay, so we had talked in class on Tuesday, Wednesday, about a few things. We talked about just some of the lingo, right? We talked about a gene. Remember, we said it's a section of DNA that codes for something, right? You have genes for all the different traits you have, okay? And then we talked about alleles. Alleles are the different forms of a gene, and so you could have the gene for eye color, but then you have several alleles for that gene. You have blue eyes and brown eyes and green eyes and hazel eyes. Those are all the different alleles, okay? And when we're talking about genetics, we always represent alleles with a letter, like a capital A or a lowercase b or a capital F or let's say a lowercase g, any of those, right? Those are all alleles. You always have two alleles. Remember, your mom is always going to give you an allele, and your dad is always going to give you an allele for every single gene in your body, okay? Mom will give you DNA to code for it. Dad will give you DNA to code for it. Now, how do you decide which of these alleles is going to win and produce whatever trait we're looking for in the person? It depends on whether those traits are dominant or recessive, right? If a trait is dominant, we always show that dominant allele as a capital letter, like maybe a capital B. If the allele is recessive, we always show it as a lowercase, okay? The capital just indicating it's bigger, it's more dominant, all right? And we also said this, dominant alleles will get expressed and dominate over the recessive allele if there's two of them. So if you had a capital and lowercase b there, whatever the capital B says, maybe it's brown hair or something, that's what is going to get coded for, and that's what color hair you're going to have, okay? Again, the only way you could express a dominant allele if you have two of them, right? If you have just one of them, right? But if you have two recessive alleles, this is the only way you'll express a recessive trait. Remember, you always get one from mom, one from dad for every single trait in your body, okay? And so knowing that then, let's kind of take that and apply that a little bit here. So we talked about what do we call those different letter combos? Well, those letter combinations are called genotypes. Okay, how you look because of those letters is called your phenotype. You can be homozygous dominant, two capital letters, homozygous recessive, two lowercase letters, or heterozygous, which is one of each. Okay. Now, let's move on. We talked about Gregor Mendel. We talked about cross fertilization. Let's talk about monohybrid crossing. So dihybrid crossing is pretty complicated. You see that's the second one down there. That's crossing two different traits. That creates a lot of possibilities. We're going to look at monohybrid. Mono meaning one. We're looking at one trait. Okay. Well, one trait. So remember, you always have two alleles. And so mom will always give you an allele and dad will always give you an allele, right? And so we're looking at one trait, one set of alleles here, okay? And so let's take a second look at this. And here's just a little bit of terminology things. If you talk about the P generation, that's the parents. The F1 generation is the offspring. And then the F2 is the offspring of the offspring. Or, think of parents as like grandparents. This would be the parents. This would be like the kids. Or this would be like parents, kids, grandkids kind of thing. Okay. All right. So let's try a monohybrid cross. It's a lot like the foiling method. You're basically looking at what are the possibilities you could get based on what the parents have. And we use a Punnett square to show that. So if you look here, one of our parents is heterozygous, big A, little a, and so is the other one. They're also big A, little a. Okay. And so we basically want to say, all right, if these are our parents, what are the possible combinations you could get, right? What are the different possibilities your kids could have? Well, when we pass these things down, this A, you could get this A from mom, and it could pair with this A from dad, and you get one of these, right? This A from mom could pair with that little a from dad, and you could get one of those. This little a from mom could pair with that big A from dad, and you get one of those. Or this little a from mom could pair with that little a from dad and get one of those. Those based on mom and dad's letter combos, these are the possibilities you could get. Okay. Now you'll notice one of them was big A, little a, and the other one was this and this, little a, big A. We always write the dominant letter first. So we would flip those around like that. Okay. So now we show the same thing with a Punnett square, right? This is how these work. They're kind of an easy way to foil, and I will want you doing these. Some of you are going to get really good at doing that in your head, and you'll not need a Punnett square. But I can't grade what's in your brain. I need to be able to see what's on paper. So make sure you're doing a Punnett square. Okay. So this is how a Punnett square works. You put one parent across the top and one parent down the side. And you make a little window, right? 4 little squares essentially is what you have. Make a big box, put a big plus sign in it, okay? And then you just basically meet up or write where those things would meet up. So this big A and this big A meet up here, and you get big A, big A. In this box, you get big A and little a. Down here, you get big A, little a meeting right there. And lastly, you get little a meeting right here, which is exactly what we said over here, right? One big A, two big A, little a's, and one little a, little a. Okay, so we're just going to use a Punnett square to show all the different possibilities here. All right, so let's do an example. All right, so it says brown eyes are dominant to blue eyes. I'm going to start with that. I will always ask you guys to make a key. So in this case, it says brown eyes are dominant. You can use any letter of the alphabet you want. There's ones I would stay away from, ones where the capital and lowercase looks exactly the same. Like I wouldn't use capital C and lowercase c. Those look too close. It's hard to tell. Okay, so use letters that are easy to make sense of. So things like, let's just use B, right? You're always going to have a dominant letter and a recessive letter. It says brown eyes are dominant to blue eyes. So my capital letter is going to be brown eyes, because brown eyes are dominant to blue eyes. Right? And my lowercase letter is going to be blue eyes. Okay? So it says if a homozygous dominant mother, homozygous dominant, well, homo means same, dominant means capital, so they must both be capital letters for the mom's genotype. Remember, that's her letter combination. So she's going to have 2 capital B's, right? And dad is heterozygous. Hetero means different, so he's going to have 1 capital, 1 lowercase. That is heterozygous, okay? So once I know my parents' genotypes, I can do my Punnett square. Come down here, draw my little window. I'm going to put 1 parent across the top. Let's go mom here, and 1 parent down the side, okay? In this first box, I'm going to get 2 capital B's. Second box, also 2 capital B's. In the third box, I'm going to get a capital B and a lowercase b. And this fourth one, I'm going to get a capital B and a lowercase b. Okay? Now, you always write your most dominant genotypes first in the offspring. Again, these are statistics, not guarantees. Okay? This is what should happen. Just like if you flipped a coin, half the times it should land on heads and half the times it should land on tails. Does that mean a coin can't land on heads 4 times in a row? No, it does not. Statistics aren't guarantees. These are statistics. This is basically telling you if you were to have children, this is the likelihood that they would have this or have that. Okay? So the genotype, my most dominant genotype is this capital B, capital B. I've got 2 of those, so I'm going to write 2, big B, big B. And then my next most dominant genotype is this one, big B, little b. I have 2 of those, 2 big B, little b's. Okay? Now, how are those kids going to look? Remember, that's phenotype. Phenotype is how you look. What color eyes are you going to have? Well, big B says brown eyes and it's dominant. You have 2 of those. Those 2 kids are going to have brown eyes. These 2 right here. But what about these 2 kids down here? Right? Big B says brown eyes. That's going to cover up that little b, remember? And so these two are going to have brown eyes, but these two are also going to have brown eyes. And so what we get here is we have 4 kids with brown eyes, right? So we're going to combine those, right? These 2 kids have brown eyes. Oops. Right? These 2 kids have brown eyes. These 2 kids have brown eyes, and brown eyes. You end up with basically a guarantee that all of your kids. So basically what this says is 4 out of 4, right, opportunities or possibilities based on our Punnett square are going to show up with brown eyes. Basically 100%, right? 4 out of 4, okay? My genotype ratio, right? We're going to basically work our way from the top and work down. My ratio is for every 2 with big B, I should get 2 kids with big B, little b, 2 to 2 ratio. My phenotype ratio is how you look, right? For every 4 kids with brown eyes, I should get 0 kids with blue eyes. Now, again, this is basically a hypothetical if you had 4 children, but you can apply those statistics no matter what, right? If you had 1 kid, you could say 50% of the time they're going to be big B. 50% of the time they're going to be big B, little b. You have an equal shot, right? If you have 1 child, you have an equal shot of being homozygous dominant or heterozygous. But in terms of eye color, we just saw if you're that genotype and these are your phenotypes, you're guaranteed to have brown eyes, right? Even though dad up here has a blue-eyed allele. He only has 1, and so that's how that works. What are the chances that they have of having a brown-eyed children? 100%. What's the percent chance they'll have a child with blue eyes? 0%. Can't happen based on mom and dad's genotypes, okay? All right. Let's try another one. So again, it says brown eyes are dominant to blue eyes. I'm going to use the same key. Big B is going to be brown eyes. Little b is going to be blue eyes. Okay. It says a heterozygous mother, heterozygous mom, meaning she's big B, little b, and a heterozygous father, meaning he's also big B, little b, and they have a child. What are the possible genotypes and phenotypes? Okay, well, let's take a look. So mom and dad are both heterozygous. We'll put mom's genotype across the top here. Dad's genotype down the side. First box is going to be big B. Second one is big B, little b. Third one is big B, little b. And fourth one is little b, little b. Okay, my most dominant genotype is the all capital one here, right? So I'm going to get 1 big B. My next most dominant genotype is big B, little b. I've got 1 here, 2 here. So I'm going to write 2 big B, little b. And then I've got 1 little b, right? I went from most dominant to least dominant when I wrote those, okay? My genotype ratio is for every 1 homozygous dominant, I should get 2 heterozygous and 1 homozygous recessive, okay? My phenotype, or how my kid's going to look, well, these first kids, they have a capital B and a capital B. They're definitely having brown eyes. These two kids here, or these 2 possibilities, right? Remember that capital B is going to cover up that lowercase b. Well, that's going to total then 3 with brown eyes. It's kind of like in math where you have to simplify. Same thing. If the phenotypes are the same, you need to combine them, right? Even though the genotypes are different, homozygous dominant here and heterozygous here, they're going to result in the same phenotype. So we have to show that, okay? And then I should have 1 down here with blue eyes, right? Little b. I'm going to actually get blue eyes out of this option, right? And my phenotype ratio then is for every 3 kids I have with brown eyes, I should get 1 with blue eyes, or essentially, of my 4 possibilities, 3 of them, right? 3 out of 4 should be brown, which is 75%. So basically what that's saying is I have a 75% chance of having a child with brown eyes, and 1 out of 4, or 25% chance of having a child with blue eyes. So it's kind of interesting. Both mom and dad were heterozygous and had brown eyes, but they could technically have a blue-eyed baby. They actually have a 25% chance down here of having a blue-eyed child. Okay. And so sometimes we can see stuff like that where neither parent shows us a particular trait, like blue eyes, but all of a sudden it pops up in the offspring. They must have been carriers for that blue-eyed allele. Okay. So I wanted to make sure we had a chance to go through a couple examples. I'll be in class if you have questions next week, but you know what you're doing in class today. Good luck, and if you have questions, let me know. Otherwise, we will see you guys later.